Okay, well, thank you very much, Michael, for that introduction. And thank you to the organizers for giving me the opportunity to speak with you today about my lab's role in Project Hope. Project Hope is a nationwide collaboration. It involves two teams, the Hope team focused on pediatric specimens and the Care team focused on adult high-grade glioma specimens, everything high-grade glioma. The coordinators are Al Young, David Aaron, Kenneth Shaw, and Mike Prados that we just heard from. And my lab is one of four labs who's doing library preparation for single nucleus RNA sequencing from frozen archival high-grade glioma specimens. And we're focused on the pediatric cohort. All of the labs involved in this exciting collaboration have one thing in common in that we've all been early adopters of the single cell sequencing technology. When my lab first adopted this technology, there was a prerequisite that you had to work with fresh tissue. And although it was very exciting to be able to profile tissue specimens at single cell resolution for the first time, we were somewhat limited in our rate of accrual, and we weren't able to get the sample sizes that we would have liked in order to draw rigorous conclusions. Also, one thing that the main thing that we're focused on in this project is looking at longitudinal specimens, looking at the evolution of glioma under therapy. And that's something you can really only do if you have access to profiling archival specimens. More recent developments from coming out of Aviv Rajev's lab, Luciano Martellotto, and others at the Broad Institute and at other institutions have adapted methodologies for extracting single nuclei from frozen tissue for use in single cell applications. So that allows us to take a frozen tissue specimen, dissociate it, extract intact nuclei, and then profile each nucleus at the RNA level, at the DNA level, looking at epigenetic modifications, or even doing single cell multiomics. And in this project, we've begun with single cell RNA-seq with an overarching goal of understanding the evolution of glioma under therapy using these cutting edge modalities. So the Project Hope's goals are to do just that, profile a cohort of longitudinal specimens via single nucleus RNA sequencing. And the focus has been on paired primary and matched recurrent specimens treated with standard of care therapy, but also on hard to acquire samples, in particular a cohort of diffuse midline gliomas. And we're looking at two different sequencing modalities, one from 10x Genomics and one that's based upon the SmartSeq2 platform. The former allows you to profile a very large number of cells, thousands of cells, at relatively low coverage, while the latter allows you to profile only a few cells, but you get very high coverage per cell. In particular, the 10x Genomics approach is really intended just to profile the 3' end of the gene, and it allows you to assess gene expression. You can look at expressed mutations to some extent, or other types of readouts, but it's not really designed for that. If you want to look at alternative splicing, if you want to look at expressed point mutations and some of these other assays, then it's beneficial to have this SmartSeq2 approach, because there you profile the full transcript and you sort of give up profiling more cells by getting deeper coverage of those individual transcripts. And so we've done a mix of both in this project. My lab profiling specimens using the 10x Genomics platform, Mariella Philbin profiling specimens using the SmartSeq2 approach. And we want to answer the question, what is the effect of standard of care therapy on tumor composition at recurrence? What about the tumor microenvironment? How does that evolve under therapy? And this really represents a true gap in our knowledge. The vast majority of clinical trials enroll patients at recurrence. However, the majority of preclinical animal models are models of primary disease. And most or many genomic studies and other profiling efforts have largely focused on specimens from primary disease. And where recurrent disease is assessed, there isn't this connection to the primary disease. There isn't this paired patient specimen aspect. So this really fills a gap in our knowledge. So why profile at the single cell level, and why profile at the RNA level? What does the RNA readout give you that you don't get from the DNA level readouts? This is a slide from a recent editorial by Howard Fine in Cancer Discovery, which I think illustrates the point quite nicely. You can see in the top panel, panel A, what it describes is the mechanisms by which genetic intra-tumor heterogeneity could potentially contribute to treatment resistance. And in the bottom panel, what he's calling epigenetic, but what I would think of as cell phenotypic heterogeneity, different cell types within the tumor microenvironment, how they might contribute to treatment resistance. In terms of genetic resistance, you can imagine a therapy being applied, such as an EGFR inhibitor. And there's one particular clone which maybe has a mutation in EGFR, which allows it to escape therapy. That clone could then grow out to recapitulate the tumor at recurrence. So that could be one genetic mechanism. Or the tumor may be evolving in sort of a Darwinian selection taking place between the individual cells, and therapy in that context may induce de novo mutations, some of which may be selected for at recurrence, which would then grow out. So these are mechanisms by which genetics, intratumor heterogeneity in genetics, can be a driver of treatment resistance. But what about the cellular phenotypes? A similar phenomenon could happen there as well. Looking at the context of adult glioblastoma, we know that there are distinct cell types within glioblastoma. There's evidence for plasticity between these different cell types or states. And in the same way that therapy might select for a particular genomic alteration that provides resistance to treatment, there may be certain cell types which are intrinsically more resistant to therapy. Those cell types may then grow out and recapitulate the recurrent tumor in the same way, independent of genotype. So two different cell types with the same genetics, one being more fit or more capable of resisting therapy, or again, a plasticity, so a shift from one phenotype to another. And we see that actually, we see a so-called proneural to mesenchymal shift in the data, and I'll describe that a little bit further in a moment. What about the evidence for genetic selection pressure under therapy? In pediatric tumors, I think the question, I don't know of a more direct answer to the question, but in adult glioblastoma, there has been some recent studies from the GLASS Consortium, which has addressed this question very directly. So this is a paper in Nature from Roel Verhoek's group, where they sequenced 288 patients treated in 35 different hospitals at the DNA level, and they found very little evidence for genetic selection pressure. In the left panel, we have one metric of potential genetic selection pressure, the ratio of synonymous mutations to non-synonymous mutations. The ratio of non-synonymous to synonymous. If a mutation is non-synonymous, then it changes the amino acid residue, and that modified protein product would potentially have a phenotype. If it's synonymous, then the protein product doesn't in principle change and wouldn't be expected to have a phenotype. So if you see an overrepresentation of non-synonymous mutations, that's evidence of a genetic selection pressure. They didn't find any evidence for that, however, in mutations which were private to the primary disease or the recurrent disease. The only selection pressure was evidenced in the mutations which were shared between primary and recurrent. In panel D, this is another metric for looking at the same thing. Here, this is a way of looking at the relative distribution of different clone sizes. If there's no selection pressure and the clones are evolving neutrally under neutral evolution, then you would expect them to roughly grow at the same rate, and they should be roughly of equal sizes. This distribution will be linear, as it is for mutations which are private to the initial disease or private to the recurrent disease. Only for shared mutations do we see evidence for a selection pressure. So taken together, these results indicate that there's only evidence of selection pressure in shared mutations, but not in private mutations to recurrence in adult glioblastoma. And so what this shows is that the clonal structure of the initial disease mostly persists into recurrence. And so there's very little evidence for selection pressure. To me, this really dovetails with the work of Christina Curtis at Stanford, where she's looked at a similar type of thing across cancers. And she has a model of Big Bang evolution of cancer. So there's an initial diversification of the genetic structure, but then those clones are remarkably stable throughout the course of the disease. Many of these mutations may be required for malignant transformation, but there's no evidence for their selection downstream during progression. I think it also dovetails with the work of Joe Costello and also from the Verhaag lab looking at regional biopsies, where again, we see very little regional variation in clonal mutation composition. So there's two sources of potential disease resistance: genetic and cellular phenotypic. We don't see the evidence for genetic selection pressure, and most genotoxic therapies which have been tried in the last couple decades have been disappointing in terms of clinical trials. So that's why we want to focus on the cellular phenotypes. How do they evolve? What are the hierarchies between different cell types? Can we understand how these cell types shift their phenotype or how they respond to therapy? And that's why one of the reasons we want to focus on using single cell technologies. Another aspect is the tumor microenvironment. Single cell technologies allow us to tease out what portion of the readout is coming from tumor cells, what portion of what we measure is coming from immune cells. Now, this is an overview of the HOPE cohort with a focus on the paired specimens. Highlighted here are the paired specimens coming from high-grade gliomas, mostly non-diffuse midline gliomas. In addition to these pairs, which we've profiled both via the SmartSig2 platform and via the 10X platform, we've also done some unpaired specimens. We have a nice cohort of diffuse midline gliomas, including specimens from the thalamus and spinal cord. And my lab, in a separate effort, has also profiled some adult and AYA specimens, high-grade glioma specimens that we're using in this analysis as well. The platform that we're using in my lab is from 10X Genomics. It's a microfluidics platform. It allows us to isolate individual cells or individual nuclei using a microfluidics chip. Together, each individual nucleus or cell is isolated in an emulsion bubble together with a capture bead. And this is a gel bead on which is immobilized capture probes. And these capture probes have been barcoded. So there's a unique molecular identifier which specifically marks just that probe. And then there's a second barcode which marks the gel bead. So these probes can capture messenger RNA. The buffer is a lysis buffer, so it lyses the cell or nucleus and exposes the RNA for capture. Once we've captured the messenger RNA, we can then cleave these probes from the gel beads and prep the library in the typical way one might prep any RNA sequencing library. But then later on, informatically, we can go back to these barcodes and tease out what molecules came from what cells and what cells came from what patients. Now, we've been innovating this platform. In addition to capturing messenger RNA, you can also capture DNA fragments. So another assay we've been developing is single cell ATAC-seq. This is an assay for transposase accessible chromatin. And what it tells you are regions of open chromatin. It tells you what transcription factors are binding where. And coupled with RNA-seq, it can give you a very rich picture of the regulatory milieu. And if we want to answer questions about cellular hierarchies within these tumors, it's extremely helpful. Single cell cut and tag is another assay that we're developing. This allows you to do transcription factor profiling or histone core modification profiling in individual cells. It's essentially the ATAC-seq assay, but instead of doing undirected tagmentation, we do antibody-directed tagmentation, allowing us to enrich for a particular transcription factor binding site. I'd like to give you some overview of the type of results we're getting. I'm going to show you data from adult glioblastomas because that's what we've, in my lab, that's the data we've actually analyzed the most at this point, and it's the analysis is mature to be able to share with you. So what are the results we're seeing in adult glioblastoma? This is a multiple factor analysis of several data sets. Data coming from my lab, all primary glioblastomas, data from Mario Suva's lab, the Neftel et al. Paper, and data from the Couturier et al. Paper. Three of the largest sources of single cell data from glioblastoma from compatible platforms produced recently. And what we find when we analyze the data using this approach, this is sort of like a principal components analysis, which can be used to combine multiple data sets from different groups, and then also assess intergroup variability so that we know that the variation we see in the results is not coming from technical differences between labs, but rather is biological. What we find is a single axis of variation between glioma neoplastic cells. Now, what we've done here, one of the great things about single cell sequencing is that we can make an effort to separate the tumor cells from the non-malignant glia, the immune cells, et cetera. And so what we're looking at here are just the tumor cells. And what we see is a single axis of variation between a so-called proneural phenotype, a cell type which is similar to an oligodendrocyte progenitor cell in terms of if it's a gene expression program, and the mesenchymal phenotype, a phenotype that's not found in non-malignant brain, a phenotype that's associated actually with the hematopoietic system, and a phenotype that's associated in glioma with treatment resistance. In particular, we see a shift towards the mesenchymal phenotype at recurrence. The mesenchymal phenotype is a hallmark of resistance at recurrence. And on average, we see a shift in neoplastic cells from the proneural phenotype to the mesenchymal phenotype. That could in principle be caused by many things. There may be changes in the cellular rates of division in the mesenchymal and proneural populations. Individual cells may be transitioning from one phenotype to another in order to escape therapy. One cell type or another may be preferentially resistant to standard of care therapy, and there's already some evidence for that in the mesenchymal population. Or genetic alterations may bias cells towards one particular cell type. In our data, we find that the first cause, the difference in proliferation, is the main source of the mesenchymal shift at recurrence. We don't see a significant increase in the total number of cycling cells, but in the cases which undergo a mesenchymal shift, we see a very marked increase in the percentage of mesenchymal cells which are cycling. In the primary disease, we and others have shown that the mesenchymal phenotype is associated with a quiescent cell, which are depleted of markers of cell cycle progression. However, following treatment, we see an activation of that cell type and a re-entry into the cell cycle of those cells based upon our sequencing, single cell RNA sequencing analysis. What about the immune milieu in these specimens? What type of things are we seeing in adult glioblastoma, and what types of analyses are we going to be performing in the pediatric specimens? Well, sorry for the busyness of this slide, but I couldn't help but share with you some preliminary data. The top component, the top half of this slide is the innate immune compartment. The bottom half of this slide is the adaptive immune compartment. In terms of the innate immune milieu, we find that the vast majority of cells, if you take a look at panel D, are in a so-called M0 unactivated state, at least according to the canonical markers. They are not activated either into a pro-inflammatory M1 state or into the so-called M2 state associated with the resolution of inflammation. That's been a hallmark for malignant progression. We do, however, see, if I can direct your attention to panel A, the bottom part of panel A, an increase in monocytic lineage cells, which are derived from peripheral blood, as opposed to CNS resident microglia, which are the other main component of innate immune cells in the tumor microenvironment. So an influx of myeloid cells from peripheral blood and an expansion of the mesenchymal phenotype, two phenomena which go hand in hand both in our data and others. I'm sort of coming to the end of the time allotted for my talk, so I'll begin to wrap up. And I'll just leave you with this brief overview of the adaptive immune compartment. What we found was that firstly, the absolute number of T cells in most of these samples are quite small. On average, about 1% of the cellular tumor are T cells. And most T cells are like in this primary case here, isolated T cells in a field of glia or confined to an apparent perivascular space. However, for some subset of the population, they have a very robust infiltration of T cells at recurrence, sort of a cohort of T cell outliers that we're looking at more closely. And for this cohort of patients, we do see robust infiltration into the cellular tumor by CD8 positive T cells. This panel I is a IHC for CD8 in this one case, which is one of the most extreme outliers in this cohort. High T cell content does correlate with survival. It seems to be driven by these outliers. And we find that hypermutation status does predict increased T cell infiltration at recurrence, a result which is somewhat at odds with recent papers by Amy Heimberger's group, Hodges et al., and Tuat in Nature 2020, but which dovetails somewhat with Wong Cancer Cell 2017, which is Rolf Verhoek's paper. We're looking at some additional novel technologies such as spatial profiling. I won't get into that now because due to time. And we're looking at ways to combine single cell sequencing with spatial profiling. Single cell sequencing tells us the cellular phenotypes. It allows us to map paracrine signaling networks. That's what we've done here, mapping the different potential signals between cell types in the tumor microenvironment. Then using the single cell data, then going to the spatial profiling to understand what's the morphological context, what's the tumor anatomical structure that these signaling networks are associated with. And lastly, validating these in a panel of patient-derived cell lines to show that these factors do in fact promote growth and proliferation, and in some contexts we can blockade them. So thank you for taking the time to listen to my presentation today. I'm very happy to answer any questions.